I'm Susan Silk from NIH, but I'm not here as myself. I'm an avatar for the people participating on the web, and so these questions have come from them. Should we be backcrossing all CRISPR-Cas9 lines to eliminate off-site targets?
Yeah, I pressed it. Okay, good. All right. So, so this is. Should we do all of them? Probably not, right? So, I think all of us up here recognize that there's already a tremendous amount of genetic variation, right? So we talk about variants versus mutants, and does that matter? Right? And it matters, I think, at the end of the day, to somebody, what I always used to call a genetic purist, right? And that's the definition of a genetic purist, is somebody whose phenotype of interest is affected by these other mutations that you're trying to remove. So I don't think we should do all of them, but in many cases it will be important to get rid of them.
Another colleague online. Oh, I'm sorry.
All right, just make a quick comment too. We can predict off-target sites based on the construction of the CRISPR. So I think it's reasonable to interrogate some number of those predicted off-target sites, at least the top 5 or 10 or something. Doesn't require sequencing the whole genome, but that's very quick and easy to do.
Another of our colleagues wants to know how are biosafety and biosecurity being addressed with the use of CRISPR technology?
What type of biosafety and security issues are we talking about here? That's a pretty broad category of.
Carlisle, I'm just reading the questions.
Okay.
Although I will say I do have an interest in this one.
Okay, so what's your interest? And then we'll
Well, OLA always defaults to the PHS policy. So our position is that we will look after the welfare of the animals as we have all of the animals, which, as you pointed out to us in the first talk, we've been selecting animals for thousands of years. So
right, and nature is producing new mutations at every meiosis and every cell division.
Our concern is for the quality of the science that we do and for the welfare of the animals, and we believe that the PHS policy adequately addresses the humane concerns of the animals.
Okay, so in terms of animal welfare, then, I mean, we have in the United States, we have the PHS policy, right? We have the, I've served as an IACUC chair, and so as someone who has extensive experience in this, every single intentional animal modification undergoes very strict review, among which is an evaluation of the possibility for affecting animal welfare. Now, in some cases, you have to balance that against the science. So we're going to produce an animal that's susceptible to cancer. Those animals are going to get cancer. So we now need to balance that model or that desired result against our perceived benefit. Right? There's always a cost-benefit analysis in each of those. But the standard biosafety in terms of animal welfare is certainly in academia or in any place where there is federal funding involved in the United States, and similar things take place overseas. I'm sure my overseas colleagues will agree that provides extremely rigorous oversight to maintain that degree of animal welfare.
If I can add to that, indeed, as Carlos says, when it comes down to animal welfare, the animals that as they are being used in the experiments, indeed, all this is already being taken care of with all the different types of laws in different countries, which there are differences, but in the end, most of them come down to the same thing. I think because the question was, I think, also about biosecurity.
It was, yes.
I think there's one exception to this so far. There might be more, and I think we're talking about that this afternoon or tomorrow morning, and that's of course the whole concept of the gene drive. And that, of course, might potentially have additional aspects for biosecurity, and I have no doubt that that will be discussed after that particular talk.
And we have one last question from online. Have any of the speakers considered developing a gene drive system for model organisms? Could a gene drive be a more efficient mechanism for producing laboratory research animals? And they ask for your thoughts on the pros and cons.
I definitely have been thinking about it, and it would save an awful lot of my budget. On the other hand, as I just said, there are a lot of other things there to take into consideration, and I'm not convinced yet if it's in the end taking everything together a good idea by itself, or yes, it's a good idea, but it has too many other potential risks. That's really a complete new way of thinking that I don't think anybody else already would get to that point at this moment.
Yeah, hi, I'm Dave Anderson from the University of Washington, but I have a question from another one of our virtual conference attendees, and I believe this is directed towards Dr. Westerfield. But the question is, can you provide any data, say for a specific gene, indicating the percentage of off-target effects which occur following Morpholino versus CRISPR technology use?
Quick answer is no, not in terms of absolute numbers. So there have been, in addition to the paper that I referred to, there are at least 2 or 3 other papers of which I'm aware that have done these comparisons of Morpholinos and CRISPRs. And I know that there are some data out there in terms of off-target effects, but I can't tell you the numbers.
Thank you.
I wanted to add a comment about the biosecurity and biosafety, because I had a similar question, and I asked that to Pilar Osorio. She's an ethicist at Wisconsin, and she was in the genomic editing meeting last week, in human genomic editing. And one of the things I think we need to remember is that today we cannot change an adult genome. This is something for the biosecurity to remember. And that, like Scott just mentioned before, we are eating foods that are already with mutations, and we have been doing it for a while, and we're not sick. So I think those are the couple of things that we need to keep in mind when we're looking at this biosecurity. And again, biosafety, there are so many regulations that institutions have about this safety and the care of the animals that we are obligated to follow. So I think we need to remember that.
Joe Newsom, University of Pittsburgh. 2 questions related to the speed of the technology that we're talking about. The first one's fairly easy, but the second one's a little complex. The first one is, if we're able in everybody's lab to create these new models very quickly and relative to how we've created them in the past, do we stop, or do we need new repositories, or stop doing repositories for embryonic stem cells and for mutant mouse models, and just everybody create it in their lab? The second question has to do with interpretation of science. As we move forward, and even with the early gene editing techniques, I've always been concerned that we don't have full phenotypic understanding of the change, and as we get more targeted, do we still, and I sort of appreciated Dr. Emborg's presentation of understanding the whole phenotype before we make any conclusions about what we're doing. I was hoping the panel might comment on those 2 issues.
So I'll, being primarily a rodent guy where the repositories exist, right? one can imagine that there will still be use for some of those, right? Mutations already exist. Maybe they would be the basis for further studies. Some of those mutations have been designed very carefully in ways that you allow various kinds of specific targeting at a particular locus or insertion of other material. But on the practice has already demonstrated that it's faster and cheaper to go and knock it out yourself and then go fish through the repository. And so in a large part, I think those things are going to be rendered obsolete. And that's personal opinion, but certainly by looking at practice in the field.
I would like to add, if you are funded by NIH, you have a resource sharing plan that includes what are you going to do with your cells that are producing, what are you going to do with the animals that you're producing. And in most primate centers, for example, they have a tissue distribution system that people can come and ask for specific tissues and obtain it, as of course there is a priority, and because there is no never-ending supply of material. It is something that is already in place, and I think can be easily translated to this new technology.
Right, so the repositories on the back end, right, there are huge numbers of repositories for genetically modified rodents, right? And I think those will continue to be important, and as the technology has improved to the point where it's fairly easy to put something down in the freezer and the costs are not that onerous to get something back out, that most strains that are developed as a research model, because most of them are developed as research models, wind up in those repositories. They're used, characterized, and then put away for potential future use, of which every so often that becomes important, right?
I think from a scientific point of view, I would prefer to stick to the same models and everybody using the same models. If indeed, as it is looking like that, it's a lot faster and a lot cheaper to simply make new models than get existing ones from the repository. Of course, we only have limited funds and limited times. On the other hand, maybe this does show that the repositories might have to start looking for ways of speeding things up so that we can maintain all those same models easily accessible to everybody who wants to use them. In the end, I think it is some sort of market mechanism.
I was intrigued by Angelika's comment about the coordination of efforts in Europe to make sure that not two labs are doing the same models. Could you expand a little more on that?
Well, this is on the large animal, and there are specific cost actions which are there that people interact, that they have courses together, that they have conferences together and work on a particular topic. And the topic of this one is particularly that we learn about the models which each one make, that we exchange materials. So we, for example, have made reporter pigs, which we then give out to other people so they can make their animal models in the reporter animals. Others are developing a repository, how to analyze large animals, how to take samples, how the samples are then processed so that the quality of the large animal samples would be similar to what you maybe get in the clinic or even better. And that people could also then come and say, oh, I need from a diabetic pig the kidney because I'm looking at this particular disease.
Hi, I'm Carl Prey. I'm an economist from Rutgers University, and so my question probably reflects some of my ignorance about the biology. But actually, I have two questions. One is someone mentioned that one of the problems with, I think it was either zinc fingers or talons, was that there were onerous patent or requirements for payments on IP on the technology, And then, but with CRISPR there is not, or with some of these newer things there is not, and I don't understand why not. Is it just because everybody's fighting about the CRISPR right now and who owns it, or you know, why the difference? And then the second question, I guess, which is more for the folks in the private sector, is whether with all this, you know, excitement about talons and CRISPR and gene editing, has that led to a big inflow of venture money, and you know, is this going to stimulate more money for research and in the future?
I'll take part of that. Well, so first off, I would say one of the big bottlenecks to the use of zinc fingers was not only the price, it was that making them was hard. So you weren't just buying access, you were buying people working and building reagents for you and distributing them to you. It was really only with the talents coming on board that you could have individual investigators have the wherewithal to effectively build editors. And of course, CRISPR's made it much easier. As I say, I can teach a 13-year-old how to make a talon. I can teach a 3-year-old how to make a CRISPR. And it's just true. But there are some issues about clarity of intellectual property. So it was always clear with the zinc finger nucleases that there was one company, it was Sangamo, that had the patent filings. They very clearly had established them as having the rights to charge those high rates, for example, and to develop products on the basis of zinc finger nucleases. The talons are pretty clear too. There were essentially two composition of matter patents that really clearly laid out who owned what. And pretty quickly, the companies that had the rights to those technologies came to accommodation with each other and have issued licenses or sub-licenses using tau effector nucleases around the world for different uses. CRISPR is, of course, total democratization. Anybody can do it. You just order it up online or the catalog, and you've got what you need to go for it. And I would say that it didn't really matter for academics anyway. No doubt, valuable products that get developed using CRISPR that are commercializable will eventually have to pay the piper. It's not clear who the piper is yet, but it's coming, no doubt. And so then it becomes, I can tell you as a business, it's cost-benefit analysis on picking which platform or the other has the clear path to market. And so, you know, we've got our toes in this for pretty much all of these platforms. We'll see how CRISPR turns out in the end. And then to answer your last question, no. No, I'd say there has been a tremendous amount of venture capital that's come into, I mean, companies virtually created overnight. No business plan, right? Just, we're going to use CRISPR to address human medicine. And, you know, you got $45 million A rounds or $25 million A rounds. And, you know, with no clear rights to the technology, it's pretty interesting to watch the speed with which this has all happened. I'm in a different space, which, you know, when you deal with food animals, all bets are off. And we're pretty excited that the aqua bounty salmon got approved by the FDA, and looking forward to hear Ron's update on that. Yeah, maybe now. We'll see. This is just technology, though. In the end, guys, these are just scissors. They're scissors and screwdrivers and hammers, and it really is about what you build with it. And so that's where the money's going to come, is what did you build? Does it have value? Essentially, we've taken the technical difficulties off the table. We can do stuff we couldn't do before, but it doesn't change the fact that it's the market that really will dictate whether a product is going to be of any utility.
David Kurtz, NIH. I wanted to put a question to you guys regarding animal numbers. And, it's still kind of questionable as this technology moves forward, whether or not we'll see an increase in animal use or decrease. I think with all new technologies, there is always, an increase in the use of resources until we understand it better. But I do feel that in the long run, that animal use will eventually go down as we understand it better. But my question now is, do you think it would be better in the initial stages to use this system in vitro and then understand the mutations that are created before you go into the animal? Because I think the concern with animal use now is you're going to generate all these founder animals, and then you have to maintain them until you understand the mutation. If you do that in cell culture, then you know what's going to happen.
I think that's a very good point. And as we already said about a couple of things, this is not really something new. We are already, in most countries, pretty strictly following the whole concept of the 3 R's, which says that if you don't have to do something in an animal per se, you're not going to get the permission anyhow. This will give extra possibilities to do in vitro simply because it will now be possible to make better in vitro models. But that will be important for some subjects, but there will be an awful lot where it's not going to make a difference. As I think Angelika said about cancer, it's a disease of the whole body. We heard the same thing about the Parkinson's. All those different things. In the end, you cannot capture that, I think, in a cultured stelline. So yes, some things can be taken out of the animal into the dish, but definitely not everything.
My lab, for example, is using IPS, like I showed you, to make neurons, and we are using them as platforms for CRISPR. And they provide insight, like you were saying, Peter, and we can figure out, okay, what kind of neurological changes I see and what molecular changes. And that is going to is informing us about what we can find in the animals. But again, will not replace the animal. I think the difference is, what is your target gene? What kind of mutation you are using? One thing is if you are making a cocktail trying to knock in genes and things like that. Another thing is, I have this disease. This is the genes that have been associated to this disease, these specific mutations, and this is what we are going to test. So it's a different perspective depending on what you are trying to analyze.
If I cannot add to that. If we could do analysis in vitro, we would also be obliged to do it. But if you, just like the example with cancer, you have the whole cancer environment, you have the whole stroma, you have an whole immune system, and all this together makes a disease. And you can't, at least at the moment, you can't replicate. There are some organ on the chips where you can do a few things, but for particular diseases, it's impossible in culture.
So that looks like the last question, and just in time, because I think everybody's probably hungry, and we want to leave ample time for lunch. But I want to end by thanking all this morning's presenters, and it's a great session. So thank you.